Okay, so this is going to be part 1 of note-taking. Day 1 of DNA, RNA, and making proteins. So write this at the top of your notes. It should be your title. If you want to change it up, that's fine. Just get the gist of it. Okay. So my first question is, how many proteins do you think every human has in their body? So in class, we had a couple different answers. Some people said 5, some people said 500, some people said 1,000, and others a bazillion. So take your guess. And we have 80,000 to 400,000 proteins in our body. That's a whole heck of a lot of protein, so my guess is they're important. So proteins are very important. Underneath your title, put making a protein, part 1 transcription. So as you can tell, there's more than one part to making a protein. The first one is transcription. So keep saying that word to yourself because it's an important word, transcription. Pretty similar to the word transcribe, which means to write. So we're making our copies and we're writing. So part 1 of transcription. This is a little GIF of it. So the purple part of the GIF is DNA. The little yellow thing is labeled as RNA polymerase. So that's an enzyme. It's a protein too. And that protein is sliding across the DNA, making mRNA. So the first little letter is pretty important. RNA is what it's making. We're going to talk about how we get from DNA to RNA, and that the whole process is called transcription. So first there's a little cartoon. This cartoon is of transcription. So the little purple dude, that is DNA. The little yellow guys, they are RNA. So the first little yellow one is saying, I'm telling you, Carol, I'm done being this guy's messenger boy. He can leave the nucleus and do it himself for all I care. So this picture tells us a lot. So the mRNA is the messenger boy to the DNA, right? That's the first thing he's saying. Then he says he can leave the nucleus himself if he wants. I don't care. Well, he can't. And the DNA is inside of the nucleus. We learned that a while ago. If there's any doubts, yes, DNA is always inside of the nucleus. If you did not know that, write that down. If you did, well, mRNA stands for messenger. That's why the RNA called himself a messenger boy. So mRNA equals messenger RNA. Okay, and transcription. What is transcription? It's the process of making mRNA copy of a gene in DNA. And this happens inside of the nucleus because that's where we find the DNA. So if we're making a copy of a gene, it's a section of the DNA. So to get to it, we got to go where it lives. DNA lives in the nucleus. I said that 3 times now, so pretty important. Write it down. Step 1, to transcription. So there's 3 steps, number 1 through 3. Leave yourself some space to write. First step is unzipping. Unzip. It's just a fancy word for opening or unfancy. So we're going to open up our DNA. DNA is a double helix. We know that. So we got to open it up and expose the bases. So those letters that you see in the little picture, those are bases. And when we unzip, the cell knows what protein it needs and unwinds that gene of DNA. So a gene is a section of DNA. That's why in the picture there's a little section highlighted. That highlighted part is open to make mRNA. Anything that's not highlighted stays closed. Step 2, base pairing. So there was bases in DNA, there's bases in RNA. So DNA is base paired with RNA nucleotides through the end of the gene. Nucleotides will always be nucleotides. Nucleotides in DNA, as we learned, is a phosphate, a sugar, and a base. RNA nucleotides are still made out of phosphate, sugar, and a base. There are some differences. We'll get to that. And in the picture, that's what we see happening. We're taking the DNA and we're making base pairs. Those are the little lines. That's why it looks like half of a DNA strand. That's why it's one strand of DNA. It's half of it. And after we're done making our copy, it's got to leave. So it's going to move out. Step 3, move it out. So the mRNA is leaving the nucleus. The purple blob is the cell. The white circle inside is the nucleus. And the mRNA was made inside of the nucleus, and now it has to leave to move on. It has to make our proteins. So at the end of the gene, RNA polymerase releases. That was the yellow guy from our GIF. That is going to go away. The DNA double helix snaps back, which just means our double strand closes again because we had it open. And our mRNA moves out of the nucleus. It's literally moving. Okay, this picture is comparing DNA to RNA. What are some similarities you see or some differences? Take a moment to look at this picture and we'll make your own observations, really. Okay, so we see guanine is on both sides. That's the green base. That's on the DNA side and the RNA side. We also notice there's adenine on both sides. That's the yellow one. They're on the DNA side and the RNA side. We see cytosine is on both sides. It's the blue one. There's a blue one on the DNA side and on the RNA side. And then we have thymine and we have a uracil. Those are not the same. They are different. So we got rid of our T's and now we have U's. So wherever you used to put a T for DNA, for RNA, now you're going to use a U. So A will pair with U, C will pair with G when we're talking about RNA. DNA is still the same. A goes with T, C goes with G. RNA, A goes with U, C goes with G. Okay? So RNA uses U instead of T. That's your new base pairing rule. So that one's important. Write it down, circle it. Super duper important. And you can also see DNA is still the double helix shape, and RNA is a single strand. Okay. Then you're going to make a table in your notes. If you're typing it, insert table. That's how you do it in Google Docs. Drawing it out, make sure you have two sides at least. First, left side should be DNA, right side should be RNA. DNA stands for deoxyribonucleic acid. You don't need to know how to spell it, but you need to know what it is. Deoxyribonucleic acid is DNA. DNA is just the short acronym for it. RNA is ribonucleic acid. RNA is just a short acronym for it. Same thing. Okay, we already said DNA is double-stranded, RNA is single-stranded. Nucleotides, we know it's made out of a phosphate, sugar, and bases. The difference is DNA has deoxyribosugars and RNA has ribosugars. So that's important to know. They're different. The sugars are different. They're bolded. Write that down. Bold it yourself. And the bases are different now too. That's why they're also colored. So for DNA we had ATGC. For RNA we have AUGC. Okay. There's also some differences in location. Where can I find DNA? We know we can find it inside of the nucleus. So if we start inside of the nucleus and we make our copies and we make our RNA and then we're going to leave, that means RNA can be found inside and outside of the nucleus. Okay? And DNA has to be bigger than RNA because RNA is only from a gene. We're making our copy from a gene, which is a section. So if our B wing is our DNA and our section is our classroom, our section has to be smaller. That's why RNA is smaller. It's a section of the DNA. And the shapes, we know DNA is a double helix. RNA has many different shapes. We're going to get into that tomorrow. Today it's just mRNA. You can draw it as a shoelace, as a string of hair, one side of a double helix. Okay, and then take a moment to pause and try to answer these questions without your notes first. If you need your notes, go ahead. You're going to draw RNA, then you're going to draw DNA. You're going to find on the picture of a cell, find the DNA and add a star to it, and where you find the RNA, add a square to it. So in other locations.